So there's orange groves. By the way, we used to have orange fights when I went to school. So some people throw snowballs in cold temperature. I threw oranges. Grapes, of course, what I'm going to talk about today. And then this is the audience participation part of the seminar. Anyone know what this is? This is what happens when you disrespect an olive tree. That's in Palm Springs, California, and I really feel sorry for that poor thing. So let's talk about grape domestication. It was domesticated about 8,000 years ago. Grapes probably once, maybe more than once. We're not quite sure yet. The thing that's really important about grapes is that they're clonally propagated. So that means they're in a permanent heterozygous state. Also, when you tend to try to breed them to themselves, they have horrible inbreeding depression. Often you try to self a grape, it does not grow. And I also told you already there's a moderate undetectable bottleneck. I won't say more about that, but it's just something that I think is fascinating about perennial domestication. It implies somehow that whoever domesticated these things gathered a lot of the genetic diversity from the wild, but how and why, I don't quite understand. Just like maize, there's been changes in the phenotype when grape was domesticated. So we have on the left the phenotypes that you're used to. On the right, we have the phenotypes in the wild. You can imagine what changed. More grapes, bigger bunches, more sugar. Those are the main things. But there is one other really fascinating thing I'll touch on very quickly. And that is the fact that actually in all of the wild Vitis, the genus Vitis, this is an old genus, by the way. There's about 70 species. They all are dioecious. There are separate male and female plants. Somehow, though, domestication broke that system, and now the grape that you and I eat and drink is a hermaphrodite. So there was a sex change during domestication, which is really interesting mystery I'll touch on, but I won't solve that mystery. So grapes are mostly in a state of eternal heterozygosity because they're clonally propagated. Some varieties of grapes have been propagated for 1,000 years, since Roman times, so that'd be even more than 1,000 years. What happens when you propagate grapes is they start to accumulate deleterious recessive variants. And you can sequence grapes and count the number of deleterious variants, again using programs like SIFT, bioinformatic methods. And what you can see, let's see here, is that if you look at a genome, so if you look at a sample of genomes, this is the range and the average number of deleterious variants in a heterozygous state. There's a lot more in cultivated than wild. So we're thinking again about this process of clonal propagation. I just want to say you can show by simulation that actually it's as you can intuit in your mind, that deleterious recessive variants can accumulate but not affect the fitness because they're recessive and they're heterozygous. So we wanted to start to look at structural variants. And again, we're interested in genome size issues, but also just sort of figuring out what's going on, how much structural variation is there. We thought grapes would be an interesting model because of this heterozygosity and also because they have a small genome size. So we went out and we sequenced the Chardonnay genome, did some high-C scaffolding, made a really nice genome. It has about 38,000 genes. Unlike maize that has about 90% transposable elements, it has about 47% transposable elements if you believe the annotations. I want you to know we did generate a circle plot. I still have no idea what it means, but I'm sure it's something very important. So one of the nice things we did by using long-read sequencing and sequencing a heterozygote, which by the way hasn't been done very successfully thus far, is that you can actually build a reference genome and then map all your long reads back to it and begin to use these long reads to tell you how heterozygous a genome is, how much structural variation is there. So if you look just within Chardonnay grapes, you can do things like this. You can, for example, make a map of coverage along here of your long reads. You can also look at where the genes are. And what you can see in this diagram, and by the way, we did this in much more detail, so hopefully you'll believe me, is that you can see coverage is pretty low here. And in fact, these little bars, these colored bars, tell you about heterozygosity. And you can see that there's not much heterozygosity in this region, which implies it's hemizygous. And in fact, we can confirm that this region is hemizygous by looking at long reads. So this gene right here only has one copy in the genome, in this heterozygous genome. This gene, on the other hand, well, can I see it? There it is. It's clearly in a region of high coverage. It's not hemizygous. There's two copies. So if you do this along the whole genome and you sort of look for these hemizygous genes, it turns out about 15% of the genes are lacking a copy in one or the other heterozygous genomes. Kind of crazy. You say, dude, you're wrong. Because I looked at that and I said to my postdoc, dude, you're wrong. So what did we do? We went crazy. We sequenced Cabernet Sauvignon with our collaborators, and we looked again. About 15% of the genes are hemizygous in Cabernet Sauvignon. We compared it to the Pinot Noir reference, which was done in the old school Sanger way. What do we find out? About 15% of the genes are hemizygous. So one of the features of grapes is really high heterozygosity in terms of gene content. Don't know what that means for function yet, but I think it's pretty neat. We could also call SVs using short reads, like I showed you at the very beginning of the talk, or using long PacBio reads. We could even compare genomes, because now we actually have a sort of a draft Cabernet Sauvignon genome that we can compare to Chardonnay. And it turns out when you do these different methods, you end up with a lot of common structural variants called by all three methods. But then also you get lots of different things, and we're not really sure which of these are artifacts or not. But this has never been done in a plant genome, I think, or in any genome, perhaps. What was really nice is that when you use the long reads, we found about 75% of them you can confirm by other methods. But also, the important point is we can use these sets of structural variants that were called in common between these two methods and use those as a gold standard, and then apply that gold standard to short read sequencing and call structural variants in a big population. So, site frequency spectrum. What do structural variants look like? We had about 480,000 structural variants throughout the genome where we could look at the population frequency. Here we're looking at a population of 12 individuals in the wild. We know which structural variants they do and don't have. And what I want to point out here, it's a little hard to see, but on the left, I think that's light blue. I'm horribly colorblind, light blue, hopefully. That are synonymous SNPs. And then we have these other colored bars representing non-synonymous SNPs, duplicated regions, transposable elements, deletions, even inversions within the genome. And the thing I want you to know is you can see that in every case, that distribution is left-leaning, meaning selection tends to act against these structural variants. Maybe that's not so surprising. If we look at the cultivated sample, it looks very similar, except for one really cool thing. As a population geneticist, this is the kind of thing that, like, it keeps you up at night. You're so excited. And that is that you can see there's some sort of discontinuity in this distribution. Here we're just looking at 12 individuals with 24 chromosomes, because they're diploids. And you can see you get up to 12, and suddenly there's this dip. And hopefully you believe that dip. We can show it's a real dip. The question is, why is there a dip? It turns out that when you only have a sample of 12, you can have as many as 12 structural variants that are heterozygous in each of the individuals. But as soon as you get to 13, at least one individual is homozygous. Now you have a recessive deleterious thing that's no longer recessive. Bam, strong selection against it, gone. Cool. Population genetics, it's the best. If you look at structural variants, you can find these same things that actually, if you compare the wild to the cultivated, there's many more structural variants because they're caught. They're sort of in an eternal heterozygous state, and we think they're mostly recessive deleterious. One of the neat things you can do when you have samples now from a wild population and a cultivated population is look all across the genome, all through the 19 chromosomes, and start to ask, where are they really different, the cultivated and the wild samples? Where those genomes are really different, that's a potential place where genes are that affect domestication phenotypes, because selection has changed them so much. So you can do this for SNPs, and it's been done a ton of times. My lab was really one of the first, but it's been done much more on all kinds of species. But you can look at this thing called FST, which measures differences between the wild and the cultivated. And you can see, like, boom, there's this huge peak on chromosome 2 where there's a big difference. That peak on chromosome 2 is where the sex determination genes are. You can also see some other peaks throughout, and I'm not going to talk about those. But this is the first time you can do this also for structural variants. And what we found is maybe some peaks in here that seem to differ between the wild and domesticated samples for things like disease resistance genes. But you also see the sex determination region. There's a lot of change in this region. And also berry color. I'm going to talk about both of those things and what's happened during domestication for both of those regions. So the sex determination region, it turns out it's a region on chromosome 2 that's been mapped before. We knew from the Pinot Noir reference what that's supposed to look like. Chardonnay, we actually sequenced it really well, so we had 2 hermaphrodites